Hello everyone, I'm Wenlong from Tongji University, located in Shanghai, China. The title of my presentation is Evolution of Vesicle Release Mechanisms in Neuro-Spike Communication. Firstly, I want to introduce the concept of molecular communication. Molecular communication is a communication method that uses molecules as information carriers, as shown in this figure. The transmitter releases molecules carrying information, and the molecules reach the receiver through free diffusion to complete the information transmission. Neuro-Spike communication is a kind of communication method that includes electrical process and the molecular communication process. The transmission process of the spike on the neuron is an electrical process, and signal transmission between neurons is a molecular communication process. The spike propagates to the end of the axon and changes the membrane potential of the neuron, which makes it release vesicles. The neurotransmitters contained in the vesicle diffuse freely to reach the postsynaptic membrane, forming a new spike on the next neuron. This molecular communication process is also called vesicle release process. Vesicle release is the process that spike propagating to the end of the axon prompts the postsynaptic terminals to release vesicle into gaps. According to the number of vesicles released in response to a spike, the vesicle release process can be divided into two types, SVR and MVR. SVR is single vesicle release. MVR is multi-vesicle release. Only a vesicle is released when a spike arrives. This process is called single vesicle release process. But if more than one vesicle is released in response to a spike, this process is called multi-vesicle release process. There is limited research on MVR models, but in fact, the SVR was originally thought to release at most one vesicle for each spike, rendering synaptic transmission unreliable. MVR, which was initially identified at specialized synapses but is now known to be common throughout the brain, represents a simple mechanism to overcome the unreliability of synaptic transmission. In this paper, a realistic MVR model is represented to investigate the BR performance, the influence of parameters involved in the vesicle release process, and data rate on BR performance is investigated in detail. Then I will introduce the system model. The traditional Neuro-Spike communication model is generally divided into three phases. First, the spikes are propagated along the axon. The input signal is represented by ST in this paper. It is composed of Dirac functions. The second phase is that the presynaptic terminals release vesicles containing neurotransmitters into gaps among neurons when ST spread to the end of the axon. Also, ST is converted into a molecular concentration sequence S'(T). It is related to the number of vesicles released, Ni. The neurotransmitters spread to the postsynaptic receptor and bind to it, forming a new spike on the next neuron. The second phase is the main part of this model. It is the vesicle release process. Like this figure, a pore-based vesicle release model is utilized. Widely releasable vesicles are stored in a pore with an upper limit of Nm, which is called reservoir, or RP, while the others are contained in a larger, available pore away from the presynaptic. Once a vesicle is released, there is a vacancy in RP, and the vesicles further away from the presynaptic are refilled into RP until the number of vesicles reach a max. The release probability of a vesicle is represented by P, and the vacancy refill rate is related to tau d, where tau d is the mean recovery time of a vacancy. The number of vesicles in RP is represented by N. In order to distinguish the two models, P of the SVR model is represented by PS, and PM represents the probability of each vesicle released in MVR model. Both the expression of PS and PM are shown in this table. In SVR model, only one vesicle is released with the probability of PS in response to a spike. And in MVR model, the number of vesicles released are based to the binomial distribution NPM. We assume that the release of each vesicle is independent of each other in MVR model. The SVR and MVR model have the same vesicle recovery process. After a simple interval, the probability of one vacancy replenishment is P-rep. So, the number of vesicles recovered in TS obeys binomial distribution N max subtract N P-rep. Based on this system model, we studied the characteristics of the vesicle release process. We assume that there are N vesicles in RP for the two models at time t. In a simple period, the number of vesicles released and recovered for the two models is shown in the table. NS and NM represent the number of vesicles in RP for the two models at time t and TS, respectively. Obviously, it can be seen that Ns is larger than or equal to n subtract 1, smaller than or equal to n max. Also, Nm is larger than or equal to 0, smaller than or equal to n max. According to the calculation formula of binomial distribution probability, the probability when Ns is equal to all different values is listed below in detail. We use Pi to represent the probability that Ns is equal to n at I, and the mean of the variation number of vesicles in Ts, Es, or Ei can be calculated. Its meaning is very clear. When Ei is larger than 0, the vesicle recovery rate is greater than the rate of vesicle consumption, theoretically. In the same way, we obtain the expression of Em. It can be seen that both Es and Em are functions of n. In order to study their characteristics, the derivative functions are obtained. From their derivative functions, both of them are smaller than 0. So, Esn and Emn are both tonically decreasing functions of n. Also, from their expression, we can see that both Es0 and Em0 are positive. Es_nmax and Em_nmax are negative. Based on the characteristics of derivative functions, both Esn and Emn have unique 0 points in 0 to nmax. By solving Esn equal to 0 and Emn equal to 0, we can get the two 0 points Ns0 and Nm0. There are their expressions. The meaning is that Ns0 and Nm0 are the theoretical equilibrium points of the SVR and MVR model. When the number of vesicles reach this point, the mean of vesicle release rate and vesicle recovery rate is equal. When n is larger than Ns0 or Nm0, the model has larger vesicle consumption rate than recovery rate. Although it is a theoretical state, it has reference significance for analyzing the change rate of vesicles. In the next section, the simulation results are introduced. From the system model, the mean system parameters involved in the system are the mean recovery time of vesicles τ, the upper limit of Rp, Nmax, the symbol period Ts, and the vesicle fusion rate Av. The bit error rate performance of the two models and the different parameter conditions is investigated. The results are shown in this figure. It is easy to see. The two models have better BR performance in the system with lower tau-d, larger Nmax, smaller TS, and higher AV. And they all influence beta rate by changing PS or PM. From the expression of PS and PM, we can say when the RP have the same number of vesicles, PS is smaller than PM. However, the SVR model always have lower BR under the same conditions. The reason is that the vesicle release rate of MVR is too high, so that the number of vesicles in RP have been in a low or even exhausted state, which will cause no vesicles to be released when there is a spike. In terms of the response to a single spike, MVR has a higher accuracy rate, but for longer signals, the MVR model have a higher beta R rate, which is its disadvantage. And in the following research on vesicle release model, this is the area that needs to be improved. That's all, thank you. Welcome to raise questions about my presentation.